So this is a summary of critical step 1.1.5, accounting for food web dynamics and models to predict management outcomes. Key players or key contributors in this critical step are Ian Duggan and Brendan Hicks, and also Matt Allen. Okay, so this is a Gantt chart of the various milestones associated with the critical step. With progress, we have a number of work streams underway, which I'll talk about separately. The first one is looking at the role of mussel nutrient excretion and biodeposition as a function of food web dynamics. Secondly, we have a project on measuring cladostrum grazing rates. Thirdly, a project on common smelt and common bullion smelt nutrient excretion rates. And finally, integrating some of these measured parameters into model code for use in lake water quality modelling. And a key partner in this work is Waikato Regional Council. So some key achievements to date. We've published 2 papers on mussel work. The first was authored by Helene Sir with various co-workers, and this was partly part of her sabbatical here a couple of years ago, and it's been written up through the Lake Resilience Program. And she looked at feeding and nutrient excretion rates of Ecorodella menzii and the implications of that for lake nutrient budgets in nearshore environments. And then most recently we've published a paper in New Zealand Journal of Marine and Freshwater Sciences on mussel biodeposition and effects of different food types on that, and I'll talk a bit about that. We've had conference presentations, one at the Ecological Restoration Society conference on the mussel biodeposition work, and also one a poster presentation at the Freshwater Sciences conference in Invercargill on trophic overlap between catfish and eels in some Waikato lakes. In terms of the fish excretion rates, Brendan's had a couple of summer students involved, and they've been calculating excretion rates of phosphorus and ammonia and nitrogen for 3 native and 3 introduced fish species. We have a zooplankton grazing rate project on the books, and the modelling work has been started by Matt Allen in association with work in another project. So first of all, a bit of background about the freshwater mussel work. Freshwater mussels historically were thought to be common in a wide range of New Zealand lakes, and they still are in some locations, but disappearing from others. They can perform a variety of important ecosystem processes, such as firstly filter feeding, clearing the water column, also excreting nutrients and biodeposits from waste material that they've ingested, and of course those biodeposits are eventually deposited onto the sediments. They can also move around biodebating sediments, and the shells provide habitat, and they can also get some sustenance from deposit feeding. So it's those processes, or in the red circles there, which I'm going to be exploring, which we have explored as part of our work in this critical step. So first of all, the work done by Helene Sir. Helene looked at nutrient excretion and clearance rates in 6 Waikato and Bay of Plenty lakes, 3 as part of the Taupo Te Karapiro Hydro Lake network, and 3 in the Rotorua lakes. And she measured chlorophyll A concentrations in the water, the clearance rate of the mussels in terms of litres per hour, and the amount of ammonia and phosphate that they excreted in terms of micrograms per hour. And if we just look at these mean values here, these are the means of all lakes. You can see that on average mussels were clearing around 0.6 litres per hour from the water and excreting around 19 micrograms of ammonia per hour and around 3.88 micrograms of phosphorus per hour. So potentially when mussel beds are particularly dense, they could play a significant role in nearshore nutrient budgets, and it's the nearshore environments where a lot of these mussels tend to occur. So Helene's work was mostly done out in the field, so the field measurements. To measure biodeposition rates, we took mussels back into the laboratory, set them up in this array of tanks under different substrate types, such as natural lake bed sand and inert silica sand which had no carbon in, and fed them a range of diets from phytoplankton stock from the aquaculture industry through to a microcystis diet, a toxin producing species, through to fine kaolin clay to mimic suspended sediment. And if we take those biodeposition results, so here we can see a mussel after it's been fed one of those different diets in a bag, biodepositing. Those biodeposits were then collected and weighed, and we scale those up for, say, a mussel bed with 100 mussels per square metre, which is in the range of what we know occurs in nature, and an average mussel rate, so that's the same across all diets. We can come up with estimates of biodeposition rates for mussel beds in terms of grams per square metre per day. And the first thing I want to note is that in terms of benthic feeding, because we compared lake bed sediments with carbon incorporated and silica sand, we're able to derive an estimate for benthic feeding, so that's feeding using the foot to move benthic particles for ingestion. We can see that it's around 1.44 grams per square metre per day could be attributed to benthic feeding. Now if we assume that's constant across different food types and take it away from biodeposits produced on a diet of microcystis or suspended solids, we can see that under a microcystis diet very little extra biodeposition was recorded, whereas under a high suspended solid And if we look at some of those different substrate types, we can see visually the effects of that biodeposition. So the two top panels are lakebed substrates fed phytoplankton stock, and you can see that in the absence of mussel, nothing obvious. The white particles where mussels are present are really just pumice particles that were there anyway. They've been brought to the surface. Under a kaolin diet, which is the bottom two panels on the left hand side with no mussel, we see pretty similar sort of picture to the stock control, but if you see a mussel and kaolin, we can see that a lot of that suspended white kaolin clay has clearly been incorporated into the sediment or onto the sediment through biodeposition, and that's not just on the sediment surface. If we look vertically through the sediment profile, we can see that a lot of that fine suspended sediment has been worked into the substrate through biotubation. So these biodeposition and biotubation processes are potentially quite significant. Now we move on to look at Brendan Hicks's nutrient excretion work that he's done, building on the initial work carried out with Di Morgan in 2013 on koi carp. Brendan's had students looking at nutrient excretion by eels, catfish, goldfish and rudd, and this year we've added common bully and smelt to that suite of species, and Brendan's currently writing up this work, and the data has been included in a synthesis of global data and ecology data series. So looking first at ammonia excretion, we can see that koi carp excreted ammonia at a much higher level per gram of fish wet mass than short-finned eels. And we can see down here smelt and bullies are on a similar sort of line to the other species, but obviously they're smaller. However, if we look at ammonia, we can see that per gram of body mass, smelt were excreting more phosphate than bullies, considerably more phosphate than bullies. But similarly, koi carp works as for ammonia, were excreting more phosphate than short-finned eels. So potentially, if we can replace koi carp with eels in lakes, we could potentially decrease the amount of nutrients that those fish excrete. And of course, if we introduce smelt into a lake, then that may have counterintuitive effects on phosphate excretion, at least compared to other native species. So the next step has really been to figure out how to integrate some of these biological processes, such as mussel biodeposition and nutrient excretion, by fish into lake models. So these biological processes can be incorporated into predictive scenarios. And Matt Allen has been doing a great job recoding some of the PC lake parameters to try and bring some of these processes into play. He's been working on Lake Waihi as part of a different project, and has been using the eel and common carp, or koi carp, nutrient excretion data from this study to model a bunch of different scenarios, including pest fish removal, and also a bunch of land management scenarios to see what is potentially working, and that work is currently still in progress. So just to summarise progress to date, we've performed pretty much completed the mussel excretion and biodeposition work, and that's been written up and published. In year 3, we're looking at the bringing in the zooplankton grazing rate experiments, and also expanding some of the application of those excretion rate estimates into lake models, and potentially with another lake scenario in addition to Lake Waihi.